Swayam Prabha. Digital India. Educated India.
I'm Swati Tiwari. I'm a faculty at the School of Biotechnology, Jawaharlal Nehru University, New Delhi. And this project was supported by DTH Swayam Prabha, MHRD, New Delhi. So we are starting a new chapter on polymerase chain reaction, or in short, PCR. And we will first discuss how the polymerase chain reaction works, and then we will talk about how to design primers for PCR, and then some of the applications of PCR. So this technique was developed by Carrie Mullis, who passed away last year in August in 2019. And in one of the obituaries published by McGill University for Carrie Mullis said as a title, The Man Who Photocopied DNA. So it is a very apt title, and so I have copied the title here, because when you want to make many copies of any page in a book or any document, then you can put it in a photocopy machine and specify the number of copies you want, and voila, you have those many copies. Similarly, the PCR technique, it basically produces many copies of the template DNA as if you have photocopied that piece of DNA faithfully. So this technique was developed by Mullis in 1983, and not only it revolutionized the way biological research is done, but also it had a lot of impact on many diverse fields ranging from anthropology to forensic sciences, and has many applications which range from evolutionary studies, gene therapy, personalized medicines, food industry, and many such applications are there, and they are too numerous to even list out in a table. So the technique involves using a thermostable DNA polymerase, and it allows to make many copies of a specified DNA template in the presence of primers and nucleotides. And in 30 cycles, it can produce up to 1 billion copies of a specified DNA fragment. So let us see how this technique it evolved and how it is carried out in present time. So before we go and talk about PCR, first remember from our previous lectures, we talked about DNA-dependent DNA polymerase enzyme. And this enzyme can use a template and in presence of a primer, it can allow the amplification of this template DNA if there are DNTPs and magnesium ions are present. So this is the template DNA on the bottom, and then this is the primer that can bind in 5' to 3' direction. So this enzyme DNA polymerase has a 5' to 3' polymerase activity, and therefore it can synthesize this complementary strand to the template DNA. So this is what is shown in pink here is the newly synthesized strand. So this DNA polymerase is the key to do PCR reactions. So let us see how that happens. So the question was that can we use this DNA polymerase to amplify a DNA template virtually without any limit. So it's not that after one cycle it cannot do it. It should keep on doing this polymerization reaction again and again and again. So how do we achieve that? So the principle behind this unlimited template amplification is like this. So first you have, supposing, a double-stranded DNA template. If you denature this template by using high temperature, then both the strands are going to separate. Once the strand separates, you can allow the primers to bind to the template, and if you have two primers that can bind to the complementary strands, which is called annealing, then the polymerization or extension of the template can be made to generate a particular product. So this is the basic strategy that is used for PCR, and then you can repeat these steps, 3 steps, again and again and again in order to give many cycles of this polymerization reaction. However, if you do that, then the problem that can be encountered is that the DNA polymerase, after all, it's a protein enzyme, and as we know that if we use high temperatures, then proteins get denatured. So DNA polymerase will get denatured and it will lose its enzymatic activity. So it will get inactivated at the denaturing temperatures. So these denaturing temperature of 95, 94 degrees are used for denaturing the DNA template. So if you use such high temperatures, then the enzyme gets inactivated. So the major breakthrough in this whole idea of scientists that can we really amplify a DNA template and make many copies was the breakthrough came through the isolation of a thermostable DNA polymerase from a bacteria, Thermus aquaticus. And this thermostable DNA polymerase was called as TAC polymerase. So TAC, basically T from this thermus and AQ from aquaticus. So it was called a TAC polymerase. So let's go through some of the historical facts here, because it is interesting to know how this whole concept developed. So before 1960s, it was generally thought that no bacteria can grow in very extreme environmental conditions, like buried in ice or at very high temperatures. But then in 1969, one scientist, microbiologist, Thomas Brock and Hudson Fries, they discovered this bacteria in thermal pools that are found in Yellowstone National Park in USA. Now, because this bacteria, it grows at very high temperatures in these thermal pools, therefore its enzymes have to be stable at more than 94 degrees centigrade for extended periods of time, because that's the natural habitat for the bacteria. Around the same time, Nobel laureate Hargobind Khurana, he was developing a method for in vitro DNA amplification, and he was toying with the idea of using a single primer and a template complex and adding the DNA polymerase again and again in order to amplify a DNA template. And subsequently, in 1971, there was another method described by Klepp, which was quite similar to PCR, and it used 2 primers. Now, during this period, people were using water baths to denature the DNA template, and then cooling water baths in order to bring down the temperature for annealing the primers. But the problem was that the DNA polymerase will get denatured. But in 1976, John Trella and his student Alice Shen from University of Cincinnati, they isolated this thermostable TAC polymerase. Because this polymerase has a DNA-dependent DNA polymerase activity, and it has a high processivity that it can add 50 to 60 nucleotides per second at 72 degrees centigrade. Uh, uh, it, it is basically highest among similar enzymes from other species. So this polymerase was at that time the only polymerase, thermostable polymerase that was known. And Kerry Mullis and his team, they developed this PCR technique between 1983 to 1985 at this biotechnology firm, Cetus Corporation. And subsequently, Kerry Mullis received the Nobel Prize in 1993, and this was the first Nobel Prize given to a person working at a private corporation, a biotechnology firm. And the history of PCR was very contentious because it led to a lot of legal warfare among different companies because they were contesting the original patents that Cetus Corporation had filed, and the challenge was that they had hidden some of the things because John Trella and Alice Shen had already isolated this thermostable DNA polymerase from Thermus aquaticus. However, nothing can take away from what Kerry Mullis and his team achieved, because the major breakthrough was not only the Taq polymerase but also design and development of the machine that can actually carry out the PCR reaction. But before going to the machine, let's first talk about what are the components of the PCR reaction and how this reaction is carried out. So when you set up the PCR reaction in a tube, then these are the various components that are added in the same order. So of course you need some water, then there is a PCR buffer. Magnesium is required to make sure that the DNA polymerase remains active. And then you have all the 4 DNTPs, so deoxy, ribonucleotide phosphates, the ATP, the TTP, GTP, and CTP. So all the 4 are added in equimolar concentrations. And then you use 2 primers. One is known as a forward primer and the other one is the reverse primer. So these 2 primers are added, followed by a target DNA which has to be amplified, and in the end you add the DNA polymerase. The reason the reaction has to be set up in this particular order is that, and all this has to be done at 4 degrees centigrade on ice. The reason being that in case you add the target DNA before and then you add the primers, then the primers will start annealing to the template, and because the temperature of annealing is not right, then some mispriming can happen. Also, the primers have to be added when at 4 degrees centigrade, and DNA polymerase is added in the end to prevent any mispriming and amplification during the setting up of the reaction. And to avoid any polymerase activity, nowadays companies offer what are known as hot start DNA polymerases, and different tricks are used to make sure that the polymerase does not become active till the reaction is put into the PCR machine and the denaturation step has been initiated. So now when you set up this reaction, now you put your reactions in a machine which is known as the thermal cycler, and you can set up the program according to which this thermal cycler will carry out the PCR reaction. Now there are, depending on the experiment, there may be variations in this entire scheme of things, but this is the basic protocol here. That first step is the denaturing step, which is carried out at 94 to 95 degrees centigrade, anywhere from 30 seconds to a minute, followed by a cooling of the samples to a temperature that is fit or optimum for the annealing of the primers, both the forward and the reverse primer. So this annealing temperature, it depends on the primer length and GC content, etc., and we will come to that a bit later, but usually it is between 55 to 65 degrees centigrade. Once the primers have been allowed to anneal for anywhere from 30 seconds to 1 minute, then again the temperature is raised to what is known as the extension step, which is at 72 degrees centigrade for these thermostable polymerases. And then depending on the length of the template, the extension time can be decided anywhere from 30 seconds to a minute or more if the template is much longer. And this cycle of 94 melting temperature, annealing temperature and extension is repeated for 25 to 30 times in order to amplify the DNA template. Now, it is very critical that the temperatures are very precise, and the timing of these temperature changes is also very critical. And these thermal cyclers, they must reach these temperatures very quickly, evenly, and very accurately in order to give a proper PCR reaction. And therefore, the earlier uses of water baths, etc., were later on discontinued as a machine was developed which could quickly change the temperature and maintain that temperature very evenly across the platform in which the tubes were incubated, and it could also maintain the temperatures very accurately for extended periods of time. So these were the basic steps. So now let us see how you analyze. So once the PCR reaction 25 to 30 cycles has been carried out, then you can stop the PCR reaction. You take out the sample and you can just analyze the products by agarose gel electrophoresis, the way we described before in first or second lecture. And then you can stain using ethidium bromide or any other DNA binding dye which is fluorescent to see the product that have been amplified in the reaction. So let's see how these PCR primers are made and how they bind to the template and how this PCR reaction it gives rise to the amplicons. So as I said, there are two primers used in the PCR reaction, the forward primer and the reverse primer. And so when you write the PCR primer, then you always write them in 5' to 3' direction. So the forward primer has the same sequence as the strand which is written 5' to 3' in your template. So as you can see that it is the same sequence as this strand here, GCTA, CGG, AA. And this primer is going to bind to this complementary strand here, which is 3' to 5', because after binding, when it coats, polymerizes in 5' to 3' direction, the strand that it will generate will be this strand which is shown on the top. The reverse primer, it will bind to this lower strand here. Again, I'm sorry, it has the same sequence as the lower strand here. But when you write this primer for 5' to 3', it will be basically a reverse of this sequence here. So written 5' to 3', it will be GCG, AT, GCCCT. Whereas in this strand, it is basically the sequence is TCC like that, right? So primer sequence when written 5' to 3' will be reverse of this sequence, and the reverse primer is going to bind to this upper strand in the 5' to 3' direction, and by polymerization it is going to reproduce this strand here, which is complementary to this strand. So this is how the primers are conventionally written. So when you write and show the primer, you show the forward primer on top of the sequence to be amplified, and the reverse primer but bottom of the template to be amplified. But remember that this forward primer is going to bind to the lower strand here, and the reverse primer is going to bind to the upper strand here. So this is how I have shown in the subsequent picture the way the primer is going to bind and amplify or polymerize the template. So how do you design a PCR primer? Now it is very crucial to design a good primer. Otherwise, if your starting material is, for example, genomic DNA from which you want to amplify, if your PCR primer is not well made, it may have regions of homology to many areas of the genome. And if the annealing temperature is not used properly or not set properly, then the primers can anneal to many different locations in the genome and give rise to many spurious products which you do not want. You may get non-specific products. So the designing of the PCR primer is very crucial, and nowadays there are many softwares that are available to design the primers, but your judgment is required to understand that of the 10 or so primers that the software may suggest, which is the one that you have to ultimately pick. So it is crucial for you to understand what are the properties that you have to look for. So as I said before, the primers are always specified from 5' to 3', left to right. And depending on the application, they may be anywhere between 18 to 25 bases in length. But for in vitro mutagenesis, sometimes there are much longer primers that are made. The primer should have a GC content between 40 to 60%. Too low and it will have a very low melting temperature, which will lead to non-specific binding. And too high, then it will be very difficult to dissociate the primer from the template. And therefore, you want to keep the GC content of the primer between 40 and 60%. And it is better that the 3' of the primer ends in either a C or a G residue, because you know that in CG pairs, they have 3 hydrogen bonds, and therefore they have a higher temperature for dissociation compared to A or a T. So if your 3' is binding very nicely to the template, then you can have a stable binding. Also, one has to be very careful that the 3' end of the primer, it is exactly matching the template. So for example, if you have to do some mutagenesis and you want to incorporate one or few bases which are not exactly similar to the template, then you don't want to put those bases in the end of towards the end of the primer, and you to prevent so that the primer binds properly to the template. So you want to put at the 3' end of the primer, it has to be exact match to the template. And also in the last 5 bases from the 3' end, if there are a couple of G or C bases, then it allows better binding to the template, and this is known as the GC clamp. Other than these very basic properties of a primer, one has to avoid regions in the primer so that there is intra-primer homology. What that means is that there are more than 3 complementary bases within the primer, because what will happen that if there are complementary bases within the primer, then the primer can fold upon itself and will not be available to bind to the template. Similarly, the inter-primer homology has to be avoided. So if the forward and reverse primers have complementary sequences, then they will bind, anneal with each other and form what is known as a primer dimer and will not bind to the template. So intra-primer homology, which can give rise to secondary structure, has to be avoided. Inter-primer homology to prevent primer dimers. So these two things have to be avoided. And the melting temperature, and this melting temperature known as Tm, this is basically the temperature at which the primer will basically dissociate from the template. So this Tm, 5 degrees below the Tm is used for annealing the primer. So this Tm has to be between 55 and 65 degrees centigrade. So lower temperature will not allow the primer to bind properly, and higher temperature it will be difficult to dissociate from the template. Also, both the primers, so the primer pairs, forward and reverse primers, they should have similar Tm, and the maximum difference of the Tm between the two primers should not be more than 5 degrees. So you may not have an exact match, but within 5 degrees, if they have the Tm, then these primers will work. So these are some of the basic requirements of how to design a primer sequence. So let us see how the cycles of PCR work and how the specific template can be amplified using a pair of primers and the 3 basic steps of denaturation, annealing, and extension. So assume that this is a template DNA where the top blue line is the 5' to 3' strand, and the bottom green line is 3' to 5'. So these two strands are the template, and it's a long template, supposing it's a piece of genomic DNA or something. So the first step is the denaturation at 95 degrees centigrade in the first cycle. So the strands, they will separate from each other. And the next is the annealing temperature. So the temperature is brought down between 55 to 65 degrees, depending on the Tm of the primers, and so the primers will come and bind. So I told you that the forward primer binds to the lower strand and the reverse primer binds to the upper strand. So this is how I have shown here, but conventionally if you're writing the primers, they are written, this one will be here and this one will be here. But this is how they bind, which strand they're binding to. That is what I have shown in this cartoon. And then the third step is the extension step. So once you allow the polymerase reaction to go on, then the complementary strands will be synthesized. Now don't go by the direction of this animation here. That is just to show that something is happening here. But basically the polymerization will be from this primer going towards the left and from this primer going towards the right. So now there is nothing to stop this polymerase. It can go on for a long time till the next denaturation step starts. So you generate a long complementary strands from these two strands. So then we have to think how the specified, I'm very sorry about it, how the specific product is produced. So let us go and see what happens in the second cycle. So now remember, after the first cycle, now you end up with 4 DNA strands, right? So this becomes one pair and this becomes another pair. And so in the second cycle, you have these strands where this is where the complementary strand had started in the first cycle, and by the reverse and the forward primers. So now in the second cycle, once the denaturation is done, the strands separate from each other, the primers come and bind again. So remember that the forward primer binds somewhere here. So this is how it is going to bind, reverse primer is going to bind, and now the extension again. So now you see that because this DNA template is starting from here, so this polymerase reaction cannot go beyond that, and therefore there is a specific length of the product that is made here. Similarly, from this primer, it can go only up to here, so it can synthesize only up to this point. However, there were the original two strands which were much longer, and the polymerase reaction can make a template complementary to that template, a much longer product. So now let's see what happens in the third cycle. So now remember that you end up with 8 strands of DNA. So this is one pair, this is one pair, this is one pair, this is one pair, right? So now you have 8 of them, and you denature, the strand separation happens, the primers come and bind again, and the extension. Now you see that because in the second cycle there were 2 such templates which had a specific length, and therefore the polymerase reaction has given rise to a DNA product which is of a specified length that was desired by the experiment. So every cycle, you notice, from 2 to 4 to 8, so every cycle the amount of DNA is doubling with each cycle. And now that in the third cycle you see that the original template, which was very long, and therefore the complementary strand was also very long, this becomes a minor component of the entire reaction, and these shorter products become a major component of the reaction. So with each subsequent cycle, these shorter products will predominate the reaction, and ultimately they will overtake, and they will be so much in abundance that when you analyze you will not even see these longer products, depending on the sensitivity of your measurement of how you visualize the product. These shorter products are the only ones which will be visualized in your agarose gel. So how many copies can be produced? So as I showed in the previous animations, each cycle of the template DNA basically doubles the template DNA. And therefore, after n cycles, 2 to the power of n copies of the DNA template are synthesized. So this is shown in this table here. So you start with 1 copy of the template DNA. In the first cycle you produce 2, then 4, 8, so on and so forth, such that by the time you complete 30 cycles, you have almost 1 billion copies of this DNA template that can be synthesized. So that is a lot of DNA that can be actually made. And therefore, this PCR technology became such a handy tool in the hands of the molecular biologist initially, because remember earlier that for cloning purposes people were just using restriction enzymes. The drawback with that was that you didn't have a very precise control over where the DNA fragment that you're cloning should start from and where it should end. But because you can design the PCR primers very precisely, you can have very accurate starting and stopping sequence, where the DNA should start, where should it end, so you can use a very precise positions of that you can use for cloning and expression purposes. So here is the one example of a PCR machine that is shown here. So you see that there is a platform here with these small holes, and this is this is known as the block of the machine. And in this block, you can put either these very small uh, 0.25 to 0.5 ml tubes or strips. They are also available as a strip that you can put in these blocks for PCR amplification. And many machines now, they have a heated lid, so that if the reaction there is evaporation, then it should not condense on the top of the tubes, and so heated lid prevents that evaporation and condensation on the lid to maintain the constant reaction volumes. There is also, of course, a keypad and the display from where you can set up the program of the PCR machine. So, in general, this block is 96 tube block, but nowadays machines with 4 blocks or 8 blocks are available, and at a go you can amplify many samples at one time. But the major improvement, as I said before, for the PCR machine or thermal cycler was that a very fast temperature ramping was achieved in these machines, because the Peltier based heating and cooling was incorporated. So from 95 to lower temperature to 72 and again to 95, the temperature can be ramped up or down very quickly using the Peltier technology to heat and cool this block very evenly and uniformly and accurately. So this was a major breakthrough that the commercialization of this PCR technology made, and these machines are now practically everywhere. And you must have heard that in the current COVID-19 disease, it is the presence of virus is determined by using a special type of PCR technology, which is known as the real-time PCR. So the machine that is employed are a modified version of this basic PCR machine. Those are known as real-time PCR machines, and I will discuss that in the second lecture. Now, the problems with the Taq polymerase is that, first of all, when it is doing the polymerase reaction, it introduces errors, and it can introduce up to 1 error in 3700 bases. So if you have a long template, say supposing you have a 5 kb template or 6 kb template, then the Taq polymerase can introduce several errors. So because it has no proofreading activity, you remember that DNA polymerases, they also have 3' to 5' exonuclease activity, and so this enzyme, it lacks this proofreading activity, so it cannot correct if it has incorporated a wrong base. And also this Taq polymerase is not very efficient in amplifying those regions of the template which may be GC rich, because they tend to form secondary structure, and this Taq polymerase, it cannot polymerize those GC rich regions very effectively. So to overcome these error prone PCR by Taq polymerase, thermostable enzymes were also isolated from other species that were found to grow in extreme temperatures. And many of these enzymes, they also had a proofreading activity. So they had a 3' to 5' exonuclease activity. Some of the examples are like PFU in polymerase or TTH polymerase. However, these enzymes, they do not match with the processivity of the Taq polymerase. I told you that Taq polymerase is very fast. It can add 50 to 80 bases per minute, but these other polymerases, they are much slower, and therefore for the same amplicon template length, one needs to use much longer extension times compared to the Taq polymerase, but that is a minor price to pay if more accuracy and high fidelity is required. So when you want to clone a gene, you do not want to clone something which has some, that has errors. So the gene should be exact copy of the gene that is present in the organism. So under these conditions, you need very high fidelity enzymes so that the cloned gene is a true representation of the gene and does not contain errors that were introduced by Taq polymerase. So under these conditions, one uses more high fidelity enzymes like PFU, TTH, and there are many other enzymes that are available in the market from commercial sources. So this lists some of those polymerases, and these companies, they have done a lot of research in order to improve the efficiency of the PCR using these some enzymes. So the technique that they use is that they use a fusion of this thermostable DNA polymerase with a DNA binding protein. And this allows the better binding of the primer to the of the polymerase with the template DNA. And this makes the PCR reaction much more efficient. So there are many ways in which such fusions have been carried out, and in many cases it has not only increased the processivity, it has also increased the fidelity of the resulting enzyme. So there are all these different trade names are given to these polymerases, thermostable polymerases by various companies, and the structure with which they were fused, or what was the original enzyme. So, for example, there is Thermo Scientific provides this fusion high fidelity enzyme, which is fused with the SSO70 DNA binding protein, and it has an increased fidelity and processivity. The PFU enzyme can amplify much longer fragments, so it is the choice polymerase if you want to do in vitro mutagenesis of the cloned genes in a plasmid. So you have to amplify the entire plasmid, and therefore in those conditions the PFU enzyme is used because it can even amplify large pieces of DNA. There are many such enzymes, for example, fusion or high-proof, high-fidelity enzyme. So different companies have these different enzymes that you can buy from them in order to set up the PCR reaction, depending on what you want to do. But for very small DNA fragments, and if you just want to see whether a particular template is there or not, you can use Taq polymerase because it is the easiest and the most efficient and most processive enzyme that's around still. So coming to the applications of PCR, it is very difficult to cover all the applications of PCR technology, but what you have to understand is that there are numerous applications in fields ranging from biotechnology, microbiology, environmental sciences to medical sciences, anthropology, food industry, and so many others. So, but some of the broad applications I have listed here. So in basic research, of course, a PCR is now the technique of choice to amplify the target genes and complementary DNAs. If you want to clone the gene, you want to express a gene, and even for sequencing, and we will be covering DNA sequencing in the next chapter. But remember that DNA sequencing now uses PCR based technique. PCR is also used to carry out site-directed mutagenesis, where you can very specifically mutate the residues that you want to mutate or delete or add to the DNA template. This technique is also used to study the DNA polymorphism, and there are many techniques to study DNA polymorphism. You may have heard the name of RFLP, which stands for Restriction Fragment Length Polymorphism. And this RFLP is markers are used to look at to study the crops, which one is the improved crop and which one carries which markers and all those things. So it is very it is used very extensively in agriculture field. And DNA fingerprinting is, as you know, that this is the technique that is used nowadays in forensic sciences. And also this PCR is, based technique is used to study the gene expression in a specialized form of PCR, which is known as reverse transcriptase PCR. And to quantitate the gene expression, nowadays a quantitative RT-PCR is also done. So we will discuss that in the next lecture. Then because the PCR can pick up differences between DNA fragments, so it is used as a diagnostic technique to either detect the presence of certain infectious organisms or for detection of genetic diseases where there might be certain mutations in the organisms. So one can use PCR-based technology to detect those genetic diseases. Also, for personalized medicines, PCR-based technology can be used to select those patients which may be good candidates for some type of treatments. Also, archaeological field uses PCR-based technology to identify the remains that they find, because PCR can amplify DNA from very, very minute amounts. So you can go as low as femtogram amounts of DNA and use special tricks to be able to amplify those DNA using the PCR technology. So you know that in archaeological field you may have a very small fragment of animal or human bone or some carcass, and you can isolate the DNA from those samples and by PCR and then study what it may be. And also one can track the migration of species by utilizing the PCR with these remains that are found in different sites. So these are some of the very broad applications. We will discuss a couple of variants of PCR in the next lecture, but I'm going to suggest some references at the end, and you can read those to look at what uses of PCR are there. So to summarize, this PCR technology was developed by Carey Mullis in 1980s, mid-1980s, and this technique employs a thermostable DNA polymerase isolated from Thermus aquaticus, which is known as TAC polymerase, and it can amplify the DNA fragment in a cyclic reaction, and usually 25 to 30 reactions cycles are done for this reaction. And the drawback of TAC polymerase is that it has a high error rate because it has no proofreading activity. So because of that, there are other high fidelity thermostable DNA polymerases that are now available from commercial sources that can be used to set up PCR reactions. And this technique uses two primers, the forward and the reverse primers, which are 18 to 25 bases long sequences that can bind to the template very specifically. And they have 40 to 60% GC content and a TM of between 55 to 65 degrees centigrade. So these primers, they can be designed either manually or using some softwares that are now available. And the PCR with each cycle doubles the amount of DNA, template DNA, and therefore if there are n number of cycles, then 2 to the power of n copies of the template DNA are produced. This is a theoretical calculation. Practically, it is slightly less than this because, as the reaction proceeds, the DNTPs are consumed, and the enzyme, even though it is thermostable, repeated heating at 95 degrees results in some loss of enzymatic activity. So this is a theoretical number here, 2 to the power of n number of copies. Practically, slightly less than this is produced. And there is no doubt that PCR has revolutionized the biological research and many other allied areas, and it has numerous applications, not only for research but also a lot of applied applications, basically. For example, for personalized medicine, for crop improvement, for forensic identification of samples, etc. So here are some of the resources that you can utilize for further reading about the PCR technique. You can go to Intech Open, and they have a lot of topics which are freely available on the web, and search for PCR even if you do not note down this entire URL. You can just Google Intech Open and search for polymerase chain reaction principle and applications. There is also another source given here. And I think it's a video lecture that describes the PCR very well. And finally, there is a animation that is given by Cold Spring Harbor Lab. So you can just note down this particular URL, or you can search for cshl.edu and polymerase chain reaction in order to see this animation of how the PCR reaction works. So I hope you have noted down these resources, and even if you have not noted it down, you can just Google polymerase chain reaction, and you will find many articles, many videos, many animations that you can look at, but be careful that you are looking at an authentic information, because sometimes on Google there will be a lot of information which is not correct. So that is why I am telling you these sources. They give accurate information. So Intech Open, sfvideo.blog.core, and DNALC cshl.edu polymerase chain reaction. So you can search for these online and read these topics for further help. So this brings me to the end of this first lecture on polymerase chain reaction, and we will have another lecture in which we will discuss more specific uses or types of polymerase chain reaction.